Plant growth regulators. Coming to the topic of plant growth regulators. So in this topic, we are going to see regarding the characteristic features. So coming to the characteristic features. Plant growth regulators are small, simple molecules of diverse chemical compositions. It is not purely proteins or steroids. They could be indole compounds also. Indole 3-acetic acid, IAA. Adenine derivatives. So indole compound is indole 3-acetic acid and adenine derivatives are N6, furfuryl, aminopurine. And adenine is a purine, N6 furfuryl aminopurine. Kinetin. Derivatives of carotenoids are abscisic acid, ABA. Terpenes, gibberellic acid. And gas, ethylene gas, so Ga3. So totally in this we are going to see about indole 3-acetic acid, which is indole compound, and N6 furfuryl aminopurine, which is adenine derivative, and the next is terpenes, so which is gibberellic acid, and gases are ethylene, and carotenoid derivative is abscisic acid. Plant growth regulators are variously described as plant growth substances, plant hormones or phytohormones in literature. The plant growth regulators can be broadly divided into two groups based on their functions in the living body. Okay, how do they function? How do they give the response? One group of PGRs involved in growth promoting activities such as cell division, cell enlargement, pattern of formation, tropic growth, means phototropic, positive phototropic, negative phototropic, positive geotropic and negative geotropic, flowering, fruiting, seed formation. And next, these are also called the plant growth promoters, which will promote all these things. Once again, cell division, cell enlargement, pattern formation, tropic growth. Why I am repeating you is you will be having some point analysis. So you will be analyzing these points again, once again. That's help you for memorizing. Flowering. After flowering, we will be having fruiting. After fruiting, seed formation. Auxins, gibberellic acid and cytokinins. Auxin, gibberellic acid and cytokinin. So gibberellic acid are terpenes. Auxins are indole compounds, indole 3-acetic acid. And N6 furfuryl aminopurine, that is cytokinin. The PGR and other group play an important role in plant responses to wounds and stress of biotic and abiotic origin. They are involved in various growth inhibiting activities also, such as dormancy and abscission. PGR abscisic acid belongs to this group only. So the gaseous PGR, ethylene, could fit into either of the groups. It could be a promoter or it could be a inhibitor of growth. So but largely as an inhibitor only we will be talking about it. Once again, cytokinin, auxin and gibberellic acid. All the three will come under activators, plant growth promoters. And abscisic acid and ethylene are the inhibitors. Ethylene will comes under both categories, but majorly we talk about inhibitors only. Remember, abscisic acid is a carotene derivative. If you see here, interestingly, all the 5 major group of plant growth promoters have been accidentally discovered. All this was started by the observation of Charles Darwin and his son Francis Darwin when they observed that coleoptiles of canary grass responded to unilateral illumination by growing towards the light source. Phototropism. So, coleoptile of canary grass. So, if you can see here, they have taken experimentally, they have taken the tip and they have cut this tip and they have put the tip here. And if you can see, the growth will be towards the light, which is phototropism. After a series of experiments, it's what concluded that the tip of coleoptiles are the site of transmittable influence that causes the bending of entire coleoptile. So, auxin was isolated by F.W. Wendt from the tips of coleoptile of oat seedlings, means Avena sativa. And Bacane seedlings are foolish seedling disease are rice seedlings. One more thing you remember, the observations of the coleoptile of canary grass was done by Charles Darwin and by his son Francis Darwin. And it was extracted from the oat seedlings coleoptile by F.W. Wendt. So, Bacane, foolish seedlings or disease of rice seedlings was caused by the fungal pathogen called Gibberella fusiculae. E. Kurosawa reported appearance of the symptoms of disease in uninfected rice seedlings when they were treated with sterile filtrate of this fungus. Even on uninfected rice seedlings also we can observe these symptoms when it is infected with this fungus. The active substance were later identified as gibberellic acid. So, so Kurosawa, his name is, and F. Scoos and his co-workers observed that they, from internodal segments of tobacco stem, callus, a mass of undifferentiated cells, proliferated only if, in addition to auxin, the nutrient medium was supplemented with one of the following extracts of the vascular tissue. Yeast extract, coconut milk or DNA. So, Scoos and Miller later identified and crystallized the cytokinesis promoting activating substance that they termed as kinetin. And in mid 960, three independent researchers reported the purification and chemical characterization of three different kinds of inhibitors. Inhibitor B, abscission 2 and Darman. So later all these three were chemically identical. So one scientist has reported as inhibitor B, another scientist is reported as abscission 2, another scientist as Darman. Later all the three are proved that chemically identical, they were named it as abscisic acid. So Cousins confirmed that the release of volatile substance from the ripened oranges that hastened the ripening of the stored unripened bananas. Later, this volatile substance was being identified as ethylene, a gaseous PGR. So, let us study some of the physiological effects of these 5 PGRs. Charles Darwin and his friend Francis Darwin. This is for auxin. Auxin, you have to remember the scientist name. Charles Darwin and Francis Darwin. From canary grass. Coleoptile of canary grass. And next, F.W. Wendt. Extracted. So these substance from Avena coleoptile. Next scientist is Gibberellic acid. This was seen in Bakane seedling disease of rice. This name is Kurosawa. And next is F. Scoos and co-workers. Scoos and Miller and Scoos and his co-workers. So they observed that the growth of the tobacco stem, in addition to auxin, they extracted the yeast, coconut milk, DNA, vascular material, and later they named it as kinetin, cytokinin. 3 independent researchers, they identified inhibitor 2, abscission, inhibitor B, abscission 2, dormin, and later all the 3 are identical in structure chemically, and they named it as abscisic acid. And cousins are the one who observed from ripened oranges. The very first one we are going to see here is auxins. Auxin, from the Greek auxin, means to grow, was first isolated from human urine. The term auxin is applied to the indole 3-acetic acid. So this is from indole compounds, derived from indole compounds, and we have to remember regarding Charles Darwin and his son Francis Darwin and F.W. Went. And other natural and synthetic compounds having certain growth regulating properties. They are generally produced by growing a piece of the stem and root, from which they migrated to the regions of their action. Auxins like indole 3-acetic acid, IBA, indole butyric acid, have been isolated from plants. Naphthalene acetic acid, 2,4-dichlorophenoxyacetic acid are synthetic auxins. Indole 3-acetic acid, indole 3-butyric acid are natural. Naphthalene acetic acid and 2,4-dichlorophenoxyacetic acid. 2,4-dichlorophenoxyacetic acid are synthetic auxin. All these auxins have been used extensively in agriculture and horticulture practices. They help in initiating the rooting in the stem cuttings. Auxins. And a wide application were used for the plant propagation. Auxin promote flowering in the pineapple. And they help to prevent the fruit and leaf drop in early stages, but promote abscission on the old and mature leaves and fruits. In most higher plants, growing apical bud inhibits the growth of lateral bud. This phenomenon we call it as apical dominance. Removal of shoot tips, decapitation, usually results in the growth of lateral bud. It is widely applied in tea plantation, hedge making. So, auxins will induce parthenocarpy. Yes. Example in tomatoes. They are widely used as a herbicide for killing herbs. And 2,4-D, 2,4-dichlorophenoxyacetic acid, used to kill the dicotyledonous weed, do not affect the mature monocotyledonous plants. It is used to prepare weed-free lawn by gardeners. Auxins also control xylem differentiation and helps in cell division. Next thing is gibberellic acid is another kind of promotory PGR. There are more than 100 gibberellins reported from widely different organisms such as fungi and higher plants. They denote like GA1, GA2, GA3 and so on. However, GA3 was the one first identified one, discovered gibberellic acid, and remains most intensively studied form. All gibberellic acids are acidic. They produce wide range of physiological responses in plants. Their ability to cause to increase in the length of axis and used to increase the length of grape stalks. Gibberellins cause the fruit like apple to elongate and improve its shape. It also delay the senescence. Thus, fruits can be left on the tree longer so to extend the market period. GA3 used to speed up the malting process in brewing industry. Sugarcane stores the carbohydrate as sugar in their stem. Spraying the sugarcane crop with gibberellic increase the length of the stem, thus increasing the yield as much as 20 tons per acre. Spraying juvenile conifers with gibberellic acid hastens the maturity period, thus leading to early seed production. Gibberellic acid also promotes the bolting internode elongation just prior to flowering in beet, cabbage, and many other rosette habit. Next, it is cytokinin. These have a specific effect on cytokinin and was discovered as kinetin, a modified form of adenine or purine derivative from the autoclaved herring sperm DNA. Kinetin does not occur naturally in plants. So, search for natural substances with cytokinin like activities led to isolation of zetin from corn kernels, coconut milk. Since the discovery of zetin, several naturally occurring cytokinins and some synthetic compounds with cell division promoting activity have been identified. Natural cytokinins are synthesized in regions where rapid cell division occurs, for example, root apex, developing shoot buds, young fruits, etc. It helps to produce new leaves, chloroplast in leaves. Lateral shoot growth and adventitious shoot formation to overcome the apical dominance. Auxin will promote the apical dominance, cytokinin will decrease the apical dominance. They promote nutrient mobilization, which helps to delay the leaf senescence. And next thing is ethylene. It is a gaseous PGR. It is synthesized in larger amounts by tissue undergoing senescence and ripening fruits. Influences of ethylene on plants include horizontal growth of seedlings. However, of the axis and the apical hook formation of dicotyledonous seedlings. If you see, the dicot seedlings will be hook formation because in the dicot we don't have the coleoptile and coleorhiza as a covering. So, in order to protect the dicot seedlings, it will be growing like a hook formation. Ethylene will promote senescence and abscission in plant organs, especially in leaves and flowers. Ethylene is highly effective in fruit ripening. It enhances the respiration rate during the ripening of fruits. The rise in respiratory rate, we call it as respiratory climactic. Ethylene breaks the seed and bud dormancy. Initiates the germination in peanut seeds, sprouting of potato tuber. Ethylene promotes rapid internode and petiole elongation in deep water rice plants. It helps leaves, upper parts of the shoot to remain above the water. Ethylene also promotes root growth, root hair formation, thus helping the plants to increase their absorption surface. Ethylene is used to initiate flowering and for synchronizing fruit set in pineapple. So, flowering will be done by auxin also, even ethylene also will be doing, and fruit setting for pineapples. It also induces flowering in mango. Since ethylene regulates so many physiological functions, one of the most widely used PGR in the agriculture is ethylene. The most widely used compound as source of ethylene is ethofon. Ethofon is an aqueous solution and is readily absorbable and transported within the plant and released as ethylene slowly. Ethofon hastens the food ripening in tomatoes, apples, and accelerates abscission in flowers and fruits. Thinning of cotton, cherry, walnut. It promotes the female flowers in cucumber, thereby increases the yield. And next, finally, abscisic acid. As mentioned earlier, abscisic acid was discovered for its role in the regulating abscission and dormancy. But like other PGRs, it also has other wide-ranging effects on plant growth as well as on development. It acts as general plant growth inhibitor and inhibitor of plant metabolism. Abscisic acid inhibits the seed germination. Ethylene promotes the seed germination. Abscission will inhibit the seed germination. ABA stimulates the closure of stomata in epidermis and increase the tolerance of plants to various kind of stress. Therefore, it is called the stress hormone. ABA play an important role in seed development, maturation and dormancy. By inducing dormancy, ABA helps the seeds to withstand desiccation and other factors unfavorable for growth. In most situations, ABA acts as an antagonistic to gibberellic acid. You may summarize that every phase of growth, differentiation and development of plants, one or other PGRs have some role to play. Such roles could be complementary or antagonistic. This could be individualistic or synergy. Similarly, there are number of events in life of plant where more than one PGR interact to affect that event. Dormancy in seeds, buds, abscission, senescence, apical dominance, etc. Remember, the role of PGR is of only one kind of intrinsic control, along with genomic control and extrinsic factors. They play an important role in plant growth and development. Many of the extrinsic factors, such as temperature, light, control plant growth and development via PGR. Some such events could be vernalization, flowering, dormancy, seed germination, plant movements, etc. We shall discuss briefly of role of light and temperature, both of them the extrinsic factors, on initiation of flowering. Yeah, here is the studying of NCRT for plant growth hormones.